Hello everyone, in this video we will learn about angiosperm. So unlike gymnosperms where the ovules are naked, in gymnosperm or flowering plant the pollen grain and ovules are developed in a specialized structure called the flower. So that is why the angiosperms are also known as the flowering plants. In angiosperm the seed are enclosed by a fruit. So whatever the seed is produced, this seed is covered by a fruit. And angiosperms are exceptionally large group of plant occurring in the wide range of habitat. Their range is in size from tiny, almost microscopic like wolfia to the tall tree of a eucalyptus which is over 100-meter tall. So they are having a wide size. So they provide us food, fodder, fuel, medicines and several other commercial important products. So they are very useful for the commercial product also. So they are divided into two classes, the dicotyledon and the monocotyledons. So the dicotyledons are categorized by having two cotyledons in their seed while the monocotyledons have only a one cotyledon. So if we talk about the monocotyledon, so they are having only one cotyledon. But if we talk about the dicotyledon, they are having two cotyledons. The male sex organ in the female in the flower is the stamen, and each stamen consists of a cylinder filament which is an anther at the tip. So if we talk about the male sex organ of the flower, it is having a stamen, and this stamen having a cylindrical filament, and on the top it is having the anther. Now the anther following meiosis to produce pollen grain. So after meiosis, it will produce the pollen grain. And the female sex organ which is found in the flower is the pistil or carpel. Now pistil consists of an ovary enclosing one or many ovules. Within ovule are present highly reduced female gametophyte that we termed as the embryo sac. And this embryo sac formation is proceed by meiosis. So it is also proceed by the meiosis, and hence each of the cell which is present inside the embryo sac, it is haploid. So only n number of chromosomes is present. Each embryo sac has a three-celled egg apparatus, egg cell, and two synergid, and three antipodal cells and two polar nuclei. So these are the cell structure in the embryo sac. So it is having three-celled egg apparatus in which one is egg cell and two is synergid, and three antipodal cell which is present on the opposite side, and at the center we have the two polar nuclei. Now the polar nuclei eventually fuse to produce a diploid secondary nucleus. So whatever the polar nuclei is there, it will fuse and it will form the diploid secondary nucleus, and pollen grain after dispersal from the anther and are carried by wind or various other agents to the stigma of the pistil. So by the process of pollination, these anthers are carried by the wind or various other agencies to the stigma of the pistil. And when these pollen grains are germinate on the stigma, it will result to produce a pollen tube, and these pollen tube will grow through the tissues of the stigma and style and reaches to the ovary, and the pollen tube enter into the embryo sac where two male gametes are discharged. So whatever the pollen tube is there, it will carry the male gametes, the two male gametes, and it will transfer to the ovule. Now, one of the male gamete will fuse with the egg cell and form the zygote that we call the syngamy, and the another male gamete fuse with the diploid secondary nucleus which is present at the center to produce a triploid primary endosperm nucleus that we call the PEN. Now, because of the involvement of two fusion, this event is termed as the double fertilization. Just because two fertilization is there, one is with the egg cell and second with the diploid secondary nucleus. So that is why two fertilization is there. That is why we called it double fertilization, and it is unique in angiosperm. Now the zygote develop into the embryo which is either having the one cotyledon or two cotyledon, and the pen, it will develop into the endosperm which provide the nourishment to the developing embryo. Now synergid and antipodal cell, it will degenerate after fertilization, And during these events, the ovules develop into the seed and ovaries develop into the fruit. So whenever the fertilization occur and zygote is formed, at that time, whatever the ovules are there, it will convert into the seed, and whatever the ovary is there, it will convert into the fruit. Now whatever the life cycle is there, it is represented in the above diagram. So this is the life cycle is there, in which we can see the flower is there, and this flower will produce the anther as well as the pistil. So this pistil is having the three part, the stigma, the style, and the ovary, and the anther is having the two part, sorry, the stamen is having the two part, anther and the filament. Now when this anther will produce meiotically, it will produce the microspore mother cell. From this microspore mother cell will produce the microsporangium. After that, it will produce the microspore, and these microspore will form the pollen grain. And whatever the ovary is there, it will produce the megasporangium, that is ovule, and after that will produce the embryo sac. And whatever the male gametes are there, which is present in the pollen grain, it will transfer into the embryo sac by the pollen tube, and after that fusion will there, it will form the zygote, and after zygote the embryo will form, and this embryo will produce the again sporophyte. Now we have plant life cycle and alternation of generation. So in plant, both haploid and diploid cell can be divided by meiotic. So whatever the cell is there, whether it is haploid or diploid, it will divide only by the mitosis. Now this ability leads to the formation of different plants. So it will produce two type of different plant bodies. One will be haploid and second will be diploid. Now the haploid plant body will produce the gametes by mitosis. So whatever the haploid is there, it is also already having a n number of chromosomes, so it can produce the gamete by only the mitosis. Now this plant body represented as the gametophyte, just because it is producing the gamete, so we can say it is a gametophyte. Now following fertilization and zygote also divide by the mitosis. So whatever the fertilization is occur, after fertilization, when the chromosome number is double, that is 2n, in that case also it will divide by mitosis, and then it will produce a diploid sporophytic plant body. Now haploid spores are produced by this plant body by meiosis. So if this sporophyte is producing the spore, the or a haploid spore, in that case it will do the meiosis. Now this in turn divided by the mitosis to form a haploid plant body once again, and whatever the spore which is produced haploid, so these haploid spore again will do the mitosis to produce a haploid plant body. Now thus during the life cycle of any sexually reproducing plant, there is a alternation of generation between the gamete producing haploid gametophyte and the spore producing diploid sporophyte. So one condition occur after second one. So however, different plant groups as well as the individuals representing them differ in following pattern. So they are having a different pattern. Now sporophytic generation is represented only by the one cell zygote. That means in first type what is having, the sporophytic generation is only the one cell zygote. Other than, there is no free living sporophyte, and this sporophyte is not free living. Meiosis in the zygote result in the formation of haploid spore. So after meiosis, what is happening, the zygote will again convert into the haploid generation. Now the haploid spore divide mitotically and forms the gametophyte. Now the dominant photosynthetic phase in such plant is the free living gametophyte, and this kind of life cycle is termed as haplontic. So just because whatever the zygote is there, so this zygote is the only sporophytic generation. After that, meiosis will occur and it will produce the haploid spore. And these haploid spore, after mitosis, it will produce the photosynthetic dominant gametophyte, and this gametophyte is free living, and that is why this complete life cycle is known as the haplontic. So we can easily understand by this diagram. So in this, what is happening? In this, we can say that after syngamy, the zygote is formed, and this zygote is the only sporophytic condition. After that, the meiosis occur, and after meiosis, it will produce the haploid spore, and this haploid spore will produce the gametophyte, which is the free living and the dominant stage of this life cycle, and that is why it is known as the haplontic life cycle. Now, in second one, what is happening is the extreme in is the type of within the diploid sporophyte is the dominant photosynthetic independent phase of the plant. The gametophytic phase is represented by a single or a few celled haploid gametophyte. This kind of life cycle is termed as the diplontic life cycle. So what is happening in this? The gametophytic state is too much reduced, and that is why what is happening, whatever the sporophyte is there, it is free living, it is dominant, it is photosynthetic, and it is independent, and that is why this type of life cycle is known as diplontic life cycle. So we can easily understand by this diagram. So in this, what is happening, whatever the sporophyte is there, it is dominant, it is independent, and whenever the gametes are formed, it is very short period of time. After that, it will again combine to form zygote, and again the sporophyte is formed, and that is why this type of life cycle is the diplontic life cycle. Now in bryophytes and pteridophytes, interestingly, exhibit an intermediate condition, that means haplodiplontic. Both phases are multicellular and often free living. However, they are different in dominant phase. A dominant independent photosynthetic thalloid or erect phase is represented by a haploid gametophyte. So that means in this the gametophyte, haploid gametophyte is dominant, and it is alternate with the short living multicellular sporophyte, totally or partially depend on the gametophyte for its anchorage and nutrition. So whatever the sporophyte is there, it is multicellular, but it is short living. It is depend partially or totally depend on the gametophyte for its anchorage and for the nutrition. And all the bryophyte represent this pattern. Now the diploid sporophyte is represented by a dominant independent photosynthetic vascular plant body. This alternate with a multicellular sporophytic or saprophytic or autotrophic independent but short-lived haploid gametophyte. So in this, reverse is there. In this, what is happening? Diploid sporophyte is dominant, and whatever the gametophyte is there, it is multicellular, but it is depend on the sporophyte. Now, such a pattern is known as the haplodiplontic life cycle. Now, all pteridophyte exhibit this pattern. So, in bryophyte, what is happening? The haploid gametophyte is dominant, and in pteridophyte, the diploid sporophyte is dominant. Now, interestingly, while most algae genera are haplontic, some of them, such as Ectocarpus polysiphonia, keeps a haplodiplontic. Ficus and algae is diplontic. So in this diagram, we can easily understand the haplodiplontic. So after syngamy, the zygote will form, which is 2N structure, and from this 2N structure, it will form the sporophyte. And this sporophyte is also a 2N structure or diploid structure. After meiosis, it will produce the spore, which is haploid, and these spore will produce the gametophyte. And this gametophyte is the haploid structure, and after that it will, by the gametogenesis, again it will produce the haploid gametes. When they are fused, it will form the zygote, and again the sporophyte will form. So this type of generation is known as the haplodiplontic generation.